Hi, this is Ritik Sir, your biology SME. Welcome to the MemoNeet. And today we are going to discuss on the new concept that is anatomy of dicotyledons and monocotyledon stem. So in the last video, we actually discussed about the anatomy of dicotyledons and monocotyledons root, right? And we also gone through some of the previous year questions also. So in this video, we will be discussing about the anatomy part, especially on the both monocot as well as dicot stem. With that thing, another very important thing is today is the last day for the Elite sale as well as PYQ and Brahmastra test series sale. So you have an offer that is you are going to get all this in a very low price. So make sure that you utilize this offer. And today is the last day for the sale. Hi. So there is something called, matlab, basically what happens is in your practicals, we have this dissection. So where you have to dissect or you have to take a transfer section of a stem in order to, see its some of the parts. So when you take a transfer section of a stem, right, it could be monocot stem or dicot stem. And when we actually observe them under microscope, this is what the structure we are going to get. Okay, and this is actually there in your practicals also. So when you take a transfer section of this dicot stem, you will be able to observe this thing. So here what happens? The cell basically you will see the outermost layer, that is the epidermis. Again, the outermost layer is actually epidermis. When you see the structure, again in the structure, if you just consider this specific part, okay. So out of all this complete structure, if you just consider this specific part, this is what the overview. Okay, this is how the overview will be. So what is the outermost layer now? It is epidermis is actually the outermost layer, and there is also a hair-like growth which is actually called as epidermal hair. So after epidermal hair, we have what? Epidermis. Then these cells are actually arranged in a multi-layered. Okay, they have been arranged in multiple layers between this epidermis and between this pericycle. Okay, so between these two, that is epidermal and the pericycle, what happens? The cells are arranged in a multiple layer. Okay, it consists of three subzones. It actually consists of three subzones. One is outer hypodermis. So can you see here? Hypodermis. So outer hypodermis. Okay, three subzones. Then the outer hypodermis consists of few layers of, it actually consists of what? Few layers of coloncymatous cells. Okay, just below the epidermis. See, if this is the epidermis, just below the epidermis there is hypodermis layer, and in hypodermis layer what you will be seeing, you will be seeing some of the coloncyma cells. Okay, which provide what? Mechanical strength to the young stem. So what I said, between this epidermis and pericycle, there consists three subzones. Okay, and outer hypodermis is a layer where you will be seeing very first thing, few layer of this consist of what? Coloncyma cells. Can you see here this layer, right? So there what? Coloncyma cells here. Okay. What is the major function of the coloncyma cells now? It provides a mechanical strength to the younger tissues. So next one is all about, again it in this hypodermis. Okay, below this, so cortical layer below hypodermis, they again consist of rounded thin walled parenchyma cells now. So after the scholenchyma cells, below the scholenchyma cells, where in the hypodermis. Okay. So below the scholenchyma, you will see the second zone, that is the para. Now they consist what? Parenchyma cells, parenchymatous cells. So what with what? A small interspace, intercellular space in between the cells. So the innermost layer of the cortex is actually called as endodermis, and the innermost layer that you can see, right? They are called as what? Endodermis. In this way, three zones are present between this epidermis and pericycle. Clear? So the first one is the collenchyma, which normally gives a mechanical support. Then parenchyma, in between there is an intercellular space between. Then the last one will be the endodermis. Clear? So this is clear. Next. The cells of this endodermis are actually rich in starch grains. Okay. So what they are? They are actually rich in the starch grains. Then this pericycle, right? So it is present on the inner side of the endodermis. They are actually present in the inner side of the endodermis. See, if you consider this as an endodermis, so this if this is an endodermis, where we are actually seeing this pericycle, they are being seeing in the inner part of this endodermis. Okay. So next to the endodermic region, we will be seeing what? Pericycle, right? So after endodermics, the pericycle region can be absorbed. Now, so above the, and where actually it is above the phloem region. See, if it is an endodermis, below the endodermis and above the phloem region, this pericycle is present. And how it is? In the form of semilunar patches of Clearenchyma, semi-lunar patches of Clearenchyma. In between these vascular bundles, there are few layers of radially placed parenchyma cells, okay, which constituents, which actually have what? Medullary layer. So can you see? That means in between there is something, this spaces. So that is actually called as medullary layer. This is clear to you? So can I move to the next point? So the next point is a large number of vacuole bundles are arranged in the ring-like structure. Okay. So the large number of vacuoles bundles are actually arranged in a ring-like structure. So this ring arrangement of vacuole bundle has actually characteristics of dicot stem. So if I need to call, okay, this structure is dicot stem, how we are going to identify? By seeing a ring arrangement of this vacuolar bundles. These are vacuolar bundles, right? How they are arranged? They are arranged in a ring-like structure. Okay. So each vacuolar bundle is how they are conjoined, then they are open and with endarch protoxylem. With endarch what? Protoxylem. That means here it is. So these things, right? So they are enlarged protoxylem. A large number of rounded parenchyma cells with large intercellular space which occupy the central portion of the stem. It is constituents what? Pith region. That means this region where you can actually see the cells, the parenchyma cells are very large, and they also have large intercellular space in between them. So that region which has been covered is actually called as pith region. So now we are clear. The first point was about the epidermis. Then after epidermis, between the epidermis and pericardium, pericycle, there are three zones, that is collenchyma, parenchyma, and endodermis. So what are they? Then after that, so after endodermis, pericycle will come. So pericycle lies between the endodermis, below the endodermis, and above the phloem region. So after that, we also have, okay, cambium is, yes. So where we will be seeing where secondary growth is happens, right? So there obviously cambium will be absorbed and it will be absent in the monocot plant anyhow. So here what happens, these vascular bundles, they are arranged in ring-like structure now. That what's represent, okay, this belongs to the dicot stem or dicot plant. And at the end, you can actually see this protoxylems are also present. And at the end, we also have a large, parenchyma cells, which are large with intercellular spaces also there, which occurs in the center portion of the stem. So it is actually called as pith region. Clear with it? So after this dicot stem, now can I move to the monocot stem? Yes? Okay. So fine. Coming to this monocot stem, one thing we need to, so comparatively, dicot and monocot stem, obviously structures are different, right? So the structure is completely different. So in the monocot stem, what happens? There is sclerum chymatus hypodermis. Okay. So it has what? Sclerum chymatus hypodermis. That means they are actually made up of sclerum chyma cells, a large number of scattered vascular bundles. Okay. So here you will actually see their vascular bundles were arranged ring-like structure. But here what we are actually seeing this vascular bundles, right? They are scattered everywhere. They are not arranged in ring-like structure. Rather, they have been scattered everywhere. Okay. Each surrounded by sclerenchymatous bundle sheet, and each are being surrounded by what? Sclerenchymatous bundle sheet or sclerenchyma bundle sheet, and a large conspicuous parenchymatous ground tissue. Getting? So another one, parenchymatous ground tissues. See, the vascular bundles that I'm talking here, so they are actually conjoint and closed. So we can see here they are conjoint and they are closed vascular bundles. Then here we also have this peripheral bundles, okay, peripheral vascular bundles, what we are actually subjecting, right? They are generally very small than the centrally located ones. So centrally located ones is nothing but, so these are centrally located. So they are generally very large when compared to the vascular bundles which are located periphery region, wherever they are located. So when compared to this part, so the vascular bundle which are actually present at this part, they are very large. That is what they are trying to say. Then this phloem parenchyma is absent here. Okay. And the water containing cavity are present within the vascular bundles. The water, yes, they are present within the vascular bundles. So this was all about what? Monocot stem. So we got to know the difference between the dicot and monocot stem. Clear? So this was all about what? The transfer section of monocot and dicot stem. So once again, after completing this chapter, that is especially this topic, I'll try to discuss very few MCQs for right now. Okay. So the very first question is the vascular bundles are scattered in what? So out of this four option, I need to understand this. It is actually a direct question, right? So vascular bundles are scattered in. So where exactly this vascular bundles are scattered? Just now I have discussed it was in monocot stem, right? So whereas in dicot stem, what happened? So we were able to see like that scattering was not there. Rather, the vascular bundle was being arranged in a ring-like structure. So that's why in the monocot stem, the vascular bundles were actually scattered. So that is the right answer. So we'll move to the next question then. So well-developed pith is found in. So exactly where do we find it? Well-developed pith is actually found in the stem and root. Yes. So where? So one thing is, so we absorb a well-developed pith in monocot roots and dicot stem, or dicot roots and dicot stem, dicot roots and monocot stem, or dicot and monocot stem. So one thing we need to understand is we need to See this. So here also well developed pith was there. So in the dicot stem it was been absorbed. And in the last video, I have also said in root, monocot root, they were been absorbed. So yes, so dicot stem and monocot root is the correct answer. Moving to the next question. Colon-chymatous hypodermis is a characteristic of. So right now only I have also discussed on this specific topic, right? So what is it? So colon-chymatous hypodermis is actually a characteristic of dicot stem, right? Because in dicot stem only you'll be seeing this colon-chyma hypodermis, right? So which has been actually absorbed in the dicot stem itself. So this was all about the dicot and monocot stem transfer section and its structure and its specific function. So in the coming video, we'll be discussing about the monocot and dicot leaf part. And not only that, that is the transfer section of that part. And we are also going to solve some of the previous years and some of other questions, statement-based questions in the coming video, that is in the next part. Okay. So still that, stay tuned with this MemoNeet app and do not forget to subscribe, like, comment, and share with your friends. Not only that, very most important thing is we need to, have lot of practices since only 8 months is there for the coming NEET exam. So practice is really very important. Simultaneously with the theory, with the concept, understanding concept, solving MCQs are also very, very, very important. So one best way is you can randomly pick any textbook, right? And you can start solving the MCQs. With that, if you want to get even more number of MCQs that too relevant to the NCERT textbook, then you can actually, go through this MemoNeet app. So where you have more than 15,000 questions, in the biology section, which is completely based on your NCERT textbook, and it is based on the NTA syllabus pattern, and all the questions have been covered with a proper explanation and the diagrammatic representation. So make sure that you utilize all this in a complete way. Thank you.